At this year's meeting, I'm presenting data from a phase 1 study of a combination of an anti-PD-1 inhibitor, MEDI0680, and an anti-PD-L1 inhibitor, durvalumab, in patients with all types of cancer. Well, what we've seen is a great single agent activity with PD-1 inhibitors and similarly with PD-L1 inhibitors. There's some data showing that you can get a greater inhibition of that pathway with dual inhibitor approach. There's also some preclinical data showing that the way that tumors progress through PD-1 blockade is upregulation of CD80, which then interacts with PD-L1 and can be an escape from response. We saw some promising results in this study. There was 1 complete response and 8 partial responses for an 18% response rate. What we've seen in this study is that dosing every 2 weeks with both drugs, we've seen only 2 grade 3 adverse events, a QT prolongation and a transaminitis. No grade 4 or grade 5. There are patients with ongoing response, 8 out of the 9 patients, ongoing responders. These drugs are both dosed every 2 weeks for 1 year, and patients are on the study past 1 year, so we've seen the durability of response. There are multiple tumor types are responding. It's very promising that we have a regimen without significant adverse events. Very promising that we can give both drugs together. The durvalumab was initially given at 3 milligrams and now at 10 milligrams, which is the standard dose. And the 680 has moved up to 20 milligram dosage. This trial is now transitioned onto its phase 2 component, which is a second-line renal cell carcinoma component. It's looking at randomizing patients who progressed off of a standard first-line anti-VEGF therapy, going on to either the one arm with the PD-1 inhibitor, MEDI0680, versus the combination of MEDI0680 and durvalumab. It's a very promising combination. The future of immuno-oncology will have to be in combination regimens, and will have to be in combination regimens that are tolerable. I think this trial has met that endpoint in the phase 1 setting, a tolerable regimen, easy to give, with minimal toxicity. We've seen a complete response and partial responses. We've been able to characterize the toxicity. I look forward to having future biomarker data from this cohort and from the renal cell carcinoma cohort. I urge patients looking for a novel approach to consider this trial. It's open in multiple sites and is on clinicaltrials.gov. There's tissue evaluation, but none of that has been presented yet. And I think it's a growing field of looking to see how we can increase response in PD-L1 negative tumors or low expressors. But I also think we've sort of failed to mention that we're not getting 100% responses in PD-L1 high expressors. So combination can hopefully get us there, and possibly this combination. There's a lot of preclinical evidence to show that this is an intelligent design. So I look forward to future ESMO and future meetings where we can present some of the updated data from the phase 2 portion of the study.